We've been talking about collagen molecule. We have seen various properties of collagen. Now let's see how this molecule is synthesized. Collagen polypeptide chains are synthesized on membrane-bound ribosomes and injected into the lumen of endoplasmic reticulum as large precursors, which are called pro-alpha chains. Remember, collagen is a protein that is secreted. All proteins, when they start forming, when they start coming out of a ribosome, they are recognized by cellular machinery. The protein synthesis halts till the ribosome docks on the surface of ER. Only then the protein synthesis resumes, and these proteins are injected co-translationally right into the lumen of ER. The precursors have short amino acids, terminal signals that are required to direct polypeptide into ER, and also NNC terminals have additional propeptides. We will see what these propeptides do. In lumen of ER, selected prolines and lysines are hydroxylated to hydroxyprolines and hydroxylysines. Some of these hydroxylysines are also glycosylated. So let's look at our glycosylated. So glycosylation, as we've talked about, is basically addition of sugar molecule or a carbohydrate molecule, monomer or larger molecule, to a protein molecule or any other molecule. So these collagen molecules, first of all, they're hydroxylated and then they are glycosylated. Each pro-alpha chain then combines with two other to form a hydrogen bonded triple helix, which we have seen the structure of. And now it is called as pro-collagen. So three pro-alpha chains come together, form the triple helix. And now the triple helix is basically referred to as the pro-collagen. I'll show you a schematic of all this in the next slide. But let's just read it out so I make sure that I'm not missing out on any important information. Hydroxyl groups of amino acids form interchain hydrogen bonds that help stabilize the triple helix. Remember, in DNA, the bases have these hydrogen bonds which result in formation of double helix. Here, the hydrogen bond is formed between the hydroxyl groups of amino acids. And this hydroxylation and subsequent glycosylation is very important for the stability of collagen. And if that is disrupted for some reason, it can cause a major problem with the connective tissue. For example, scurvy, you must have heard about it. It's a disease basically which was common in sailors because they did not have enough vitamin C, which comes from fresh fruits primarily. So vitamin C or ascorbic acid deficiency resulted in these sailors, when they made their alpha chains, they were not able to form the stable triple helix and were immediately degraded. One of the symptoms of this disease is loose teeth because the collagen present in that area has a high turnover rate. So you have to keep making more collagen to keep your teeth in their sockets. So since new collagen was defective, the old collagen was no longer useful, the teeth would fall out. You might have seen that in movies where you have these sailors or pirates which have a lot of missing teeth. Collagen in bones has a half-life of 5 years, or basically takes 10 years to replace a collagen molecule. So different collagen molecules have different turnovers. Generally, most proteins have a turnover rate in hours or days at the most. But collagen present in bones has a turnover rate of 10 years. So let me show you this dramatically. Here is the ribosome. It was making pro-alpha chain. It docked on the surface of ER. It injected co-translationally this polypeptide. We call this polypeptide the pro-alpha chain. Notice that there's a green region present here. This is the region that we have said will be removed from the collagen molecule once it matures. So this pro-alpha chain, it is injected. Once it comes into the ER, here it goes through modifications. For example, very importantly, the hydroxylation of selected prolines and lysines. Here's the hydroxyl group. Some of these hydroxyl groups, especially on the hydrolysines, they will be glycosylated Here you can see this blue entity, this blue hexagon. This is basically representing a carbohydrate molecule. Once these modifications have occurred on the pro-alpha chain, 3 of these chains, they come together and they form this proto-collagen triple helix. Again, that green region that I pointed out here is the propeptide. The propeptides are removed from the mature collagen molecules. Propeptides have very important function. One, we have said that they direct this polypeptide, the pro-alpha chain, into the lumen of ER. Secondly, these propeptides also prevent the self-assembly of these collagen molecules and forming these fibrils that we have seen in the previous slide. So just imagine if collagen molecules started assembling in ER into fibrils, which can be, we have said, can be large structures. They can be several micrometers long. They can disrupt the normal functioning of the cell. So from ER, we know that molecules are packaged into vesicles. They go into the Golgi apparatus where they are enriched into other vesicles, and those vesicles fuse with the plasma membrane, and secretion is basically, cells basically secrete different molecules that way. So here that molecule, that vesicle, this is the vesicle that came from ER. It will fuse with the plasma membrane. This is the plasma membrane of fibroblast, for example, a cell which makes a collagen molecule. After the pro-collagen molecule has been secreted, there are enzymes embedded in the plasma membrane that will remove these propeptides. Once these propeptides have been removed, collagen will basically self-assemble into fibril. Again, see that classical striation. We have seen why that is. Again, I will show you more details of the striation. This fibril will again can assemble into larger structures, collagen fibers. Here I would like to mention that collagen molecules, since we have said they resist tensile forces, so they need to be linked together by strong forces, and they in fact are linked by covalent bonds. These collagen molecules, these triple helices, they have been linked together by these covalent bonds. Amount of bonding depends upon where the collagen molecule is. For example, collagen in the areas which has to resist a greater tensile force will have more intramolecular cross-linking than collagen which do not have to resist greater tensile forces. These cross-links are formed in several steps between lysines and hydroxylysines, and they basically strengthen the collagen fibrils. So collagen fibril, as I mentioned earlier, many of these will come together and form the collagen fiber, and aggregation, which is basically aggregation of fibrils forming the structure referred to as collagen fiber. Now I'm going to basically read out what I have already said. So just to ensure that I haven't missed out on any important information. I have already said this, but let me repeat this out. Read out the slide. After excretion, the propeptides of the fibrillar pro-collagen molecule are removed by specific proteolytic enzymes outside the cell. This converts the pro-collagen molecule to collagen molecules. Now they're mature molecules which assemble in the extracellular space to form larger collagen fibrils. And these propeptides have at least 2 functions, as I mentioned. First, they guide this molecule to ER, and then also they prevent a formation of these triple helices while the collagen molecules is inside the cell. Here is a disorder, osteogenesis imperfecta. This is osteogenesis imperfecta type 3. Collagen is a very important molecule. If it is not functioning properly, you can see the result. This child has disfigured bones. Again, an example of collagen disease.